Hello students, let's break this down and understand the key concepts related to biotechnology and recombinant DNA technology. Biotechnology, in simple terms, refers to the large-scale production and marketing of various products and processes by using living organisms, their cells, or enzymes. Think about things like antibiotics, vaccines, enzymes used in detergents, and so on. Now, when we talk about modern biotechnology, we are mostly referring to genetically modified organisms, GMOs. This became possible only after we learned to manipulate the chemistry of DNA. The most important point here is the development of recombinant DNA technology, also known as genetic engineering. So, what exactly is this? It's a set of techniques used to alter the genetic material, DNA, of an organism to achieve desirable traits. For example, producing insulin using genetically modified bacteria. Now, let's break down the steps involved in recombinant DNA technology. Isolation of genetic material, DNA first. We need to extract the DNA from the cells. Cutting the DNA at specific sites, we use restriction endonucleases, which act like molecular scissors. They cut the DNA at specific sequences. Imagine if you have a long string and you need to cut it precisely at certain points. That's what these enzymes do. Inserting the gene into a vector. Once we have the desired gene, we need to transfer it into a host cell. For that, we use vectors like plasmids, circular DNA molecules, or viruses. They act like delivery vehicles, ferrying the desired gene into the host. Ligation of DNA molecule. Here, we use an enzyme called DNA ligase. It works like glue, joining the cut pieces of DNA with the vector DNA to form recombinant DNA. Transformation of host cell. The recombinant DNA is then introduced into the host organism like bacteria or yeast. Expression of foreign gene. Once inside, the host machinery helps in the expression of the foreign gene, meaning it produces the desired protein or product. Purification of gene product. After production, the product, like insulin or antibiotics, needs to be purified for further use. Formulation. Finally, we prepare the purified product for marketing. Now, if we are producing something on a large scale, it requires specialized equipment called bioreactors. These are large vessels designed to provide the optimal conditions for microbial growth and production of the desired product. Why is this important? Because biotechnology has revolutionized fields like medicine, agriculture, and industry. It has allowed us to produce medicines like insulin and vaccines in large quantities, develop pest-resistant crops, and even produce enzymes used in various industrial processes. Remember, in exams, they often ask about the steps of recombinant DNA technology or the role of enzymes like restriction endonucleases and DNA ligase. So, keep this structure in mind.